Good morning, everyone. I'm Dr. Bagh. I'm MD in Clinical Biochemistry, and I'm sharing a video how to make biochemistry learn easy. Today, I'm going to talk about fatty acid synthesis, also known as de novo lipogenesis or DNL. Please know that to know any pathway in your body, you must know whether the pathway is in a high insulin state or a low insulin state. Why? Because high insulin state is known as a well-fed state, whereas a low insulin state is a state when other antagonist hormone antagonists to insulin are high, like glucagon, cortisol, and we'll talk about more later. And you should know what's the site of the pathway. Once you know the site, you should know whether the pathway is cytosolic, mitochondrial, or both. Why is this important to know? Because mature RBC lack mitochondria. So you must know which pathways in your body are the ones which are not happening in your mature RBC. Then, what's the starting molecule or the substrate of the pathway? Then, what's the purpose of the pathway? Why is it important to know purpose? Because if this pathway is shut down in your body, if you know the purpose, you will know what you will not get in your body. Then, rate limiting enzyme of the pathway. Are they very important? Yes, rate limiting enzymes are the bottleneck of the pathway. If these enzymes are blocked, the whole pathway slows down. On the contrary, if these enzymes are activated, the whole pathway can be activated. There can be more than one rate limiting enzymes in a pathway, and we must know their regulation. Then, what are the regulated enzymes? What's the difference? The rate limiting enzymes are regulated, but the regulated enzyme may not be rate limiting. So, you must know the additional regulated enzyme of the pathway. Then, energetics of the pathway. Most of the pathway give energy and or ATP in the form of ATP, or some pathways consume energy in the form of ATP. So we should know energetics too. And lastly, clinical significance, if any. Since I'm talking about fatty acid synthesis, also known as de novo lipogenesis or DNL, let's complete this list with this pathway. First of all, is this pathway a well-fed state pathway? Very well. It is a well-fed state pathway and a high insulin pathway. What's the site of the pathway? Major site of this pathway is liver, and minor site of the pathway is adipose tissue. We must know site. Yes, we must know site to know the tissue affected if this pathway, which may disrupt this pathway. Is it cytosolic? Purely cytosolic pathway. Substrate. The starting molecule of this pathway is acetyl-CoA. Now you must also know this fact. Since I told you that fatty acid synthesis is a well-fed state pathway, happens in high insulin, know that you're getting acetyl-CoA in a well-fed state or high insulin by burning carbs. So you have to burn carbohydrate to get acetyl-CoA in a high insulin or well-fed state, not fat. Then what's the purpose of the pathway? Well, you know we are talking about fatty acid synthesis. So for sure it will form fatty acid, but don't forget that fatty acid synthesis goes into TAG stores. So remember that fatty acid synthesis goes into TAG stores in your body. I will talk about that later. Rate limiting enzyme of the pathway. The rate limiting enzyme for this pathway is only one, which is acetyl-CoA carboxylase, also known as ACC. What is the regulated enzyme of the pathway? The regulated enzyme of the pathway is fatty acid synthase complex, and also known as FAS. What are the energetics of the pathway? Remember always in your body that synthesis need ATP. So this pathway will not produce ATP, but this pathway will require energy in the form of ATP. Clinical significance, if any. Yes, because fatty acid goes into TAG stores. Later on, we will see what are the clinical significance of TAG accumulation in your liver. Is there any clinical signification? Yes, TAG accumulation in the liver leads to Fatty liver. So we'll talk about those concepts of clinical significance with this pathway. This is just an overview of fatty acid synthesis, also known as de novo lipogenesis. Kindly give me your feedback regarding the overall view of the pathway, and I will soon add the video for the steps of fatty acid synthesis and regulation of rate limiting enzyme. Thank you.